Hi, I'm David Guevara, and today I'm going to talk about ProFeatX, which is a parallelized protein feature extraction suite for machine learning. Well, machine learning has been applied to many different fields, including molecular biology in areas such as genomics, proteomics, and transcriptomics, which has helped us a lot. With us, I mean scientists and bioinformaticians in general. However, machine learning has two big problems. One, it requires lots of data, and two, it requires that every input data point has the same number of values in it. So here's an example. In the image, we see that there are three protein sequences. These are amino acid sequences. But we cannot send that directly to the AI because the first sequence has 10 amino acids, the second one has 6, and the third one has 8. So what are we gonna do? We're gonna transform that so that every sequence has the same number of values. And in this example, every sequence here has 8 values. So what we did was use some kind of algorithm that helped us translate or extract 8 features from these letters. And that can be used as an input to the algorithm, to the AI. So throughout the years, researchers have come up with numerous methods to do this, with varying lengths, and there are multiple suites that compile such methods. But we did it so the number of methods was the highest, and it also would be done in a fast way. When it's just a few thousand of sequences, speed isn't that important, but some algorithms are heavy in calculations, so when you want to encode hundreds of thousands or even millions, speed becomes quite relevant. And here I'm talking from experience, because once I had to encode more than 1 million sequences and it lasted for a couple of days. And that's not what we want, right? So what we did was developing it in C++ with OpenMP to make good use of parallelization, since most current tools are developed in Python or R. We compiled 50 different descriptors or algorithms, which are available in the standalone version. I gotta say that we did a standalone version and a web version. And the web version only lets people use 31 out of the 50 due to the other 19 requiring additional files. But here you can not only encode sequences, but also protein-protein interactions. And also it lets you input DNA or RNA sequences, so the web page, using transdecoder, finds coding regions parting from those sequences and encodes those proteins that were found. In addition, we ran some tests against other suites, and as you can see here to the right, ProFeatX is the one with the highest number of descriptors with 50, while the second has 38, and also is the fastest one by more than 6 times compared to the second fastest one. Here 764 against 4560. So that's quite a big difference. We made it on a single thread. So yeah, the tests were run on a single thread to make it fair, because if they don't use parallelization, there wouldn't be much. Well, it'll be kind of like cheating. Now, in the future, we might add some more methods. Add the PPI, protein-protein interaction, and DNA or RNA functionalities to the standalone version, and also add dimensionality reduction with PCA or other algorithms suited for this task. Because we gotta check which algorithm isn't too hard to implement in C++. Because, for example, there's some autoencoder based methods which might be difficult to add. If it was in Python, then the story would be different. But here we're talking about C++, and there might not be libraries for that. And that's it. Thank you for watching. If you want to contact us, here are the links to our labs, web pages, and also our emails. Also, here's the link to the web server. And if you got any questions, you can go ahead and ask down below or wherever the question box is. Thank you.